Hello friends, welcome to Easy Nurse, where nursing education made easy. So in the series of your exam preparation, we were discussing about different subjects and their important topics. So in this session, I will be starting with anatomy, physiology and microbiology, which subject is also known as bioscience. So in this subject, we will be starting over the different topics that are very important. Okay? And I will suggest the most important question which will definitely come in exam. Okay, you can assure like at 80 whatever I will discuss, from that 80% of the topics will definitely come. Okay, so let's just start with our session with further any delay. So first we will know about the cell. Okay, so to describe about cell is actually the most singular unit of our body or of any organism. Okay, cell is the most smallest unit of any living microorganisms or any type of organism. It can be any animal, it can be any plant, or it can be any human beings. Okay, so to know about the cell, we have to know about the structure of a cell. Okay, so if we look up of the structure, what we can see? Okay, we can see a cell membrane will be there. There will be cytoskeleton which will be inside. There will be plasma, nucleus and all. Okay, so mainly the cell will be made with these things. Okay, that we will discuss it later. First we have to know is cell division. So why we have to know about the cell division? To understand how a cell will grow, or how, like if you have seen babies are growing, right? Like babies will grow excuse me, very sorry for that. Okay, so when the baby will grow, okay, जब लोग बच्चे से बड़े होते हैं, okay, not only their age is increasing, right? Each and every organ of their body, okay, each and every part of their body will grow. Okay, if they don't have any growth problems and all. Okay, so for that we really need to know about the cell division. Okay, so cell division is the process by which a parent cell, what is the parent cell? Like if a cell is there, let me show you guys. Okay, so if a cell is there, and from this cell it is dividing and making two cells. Okay, it is dividing and making two cells, then this cell will called as parent cell. Okay, from the parent cell into two daughter cell, then we will call this process as a cell division. Next we have a cell division consists of two phases. If we look, okay, that is nuclear division and cytokinosis. What is the nuclear division? See, if we look after the nuclear division, इसके दो तरह है, ठीक है, एक है माइटोसिस, एक है मियोसिस. माइटोसिस में क्या होता है, ये जो न्यूक्लियस है ना, ये सिर्फ दो हिस्सों में बांटेगा, ठीक है. लेकिन मियोसिस में क्या होता है, मियोसिस का अगेन दो फेज है, ठीक है. एक में ये दो सेल में मतलब डिवाइड हो जाएगा, और दूसरे स्टेज में वही दो सेल फिर से और दो सेल में डिवाइड होंगे. ओके, सो दिस इज द डिफरेंस बिटवीन मियोसिस एंड माइटोसिस. ओके, सो ये न्यूक्लियस डिवीजन होने के बाद ही एक सेल, ये इसको साइटोकाइनेसिस डिवीजन कहते हैं, ठीक है, इसको साइटोकाइनेसिस डिवीजन कहते हैं. तो न्यूक्लियस डिवीजन कंप्लीट होने के बाद ही साइटोकाइनेसिस डिवीजन होगा. सो माइटोसिस में क्या होता है, माइटोसिस एक टाइप ऑफ सेल डिवीजन है जिसमें दो डॉटर सेल, ठीक है, दो डॉटर सेल प्रोड्यूस होते हैं और काइंड ऑफ सिमिलर दिखते हैं. ओके, एंड दे हैव द क्रोमोसोम्स एज द पेरेंट न्यूक्लियस जब वो ग्रो करते हैं. ओके, और इट ऑकर्स इन टू प्लेसेस. फर्स्ट होता है डीएनए रेप्लिकेशन और नेक्स्ट होता है डिवीजन ऑफ साइटोप्लाज्म दैट आई टोल्ड. ओके, सो व्हाट इज द फेजेस ऑफ माइटोसिस? इट इंक्लूड प्रोफेज, मेटाफेज, एनाफेज एंड टेलोफेज. प्रोफेज में क्या होता है, प्रोफेज में ये सेंट्रियोल्स, ये जो सेंट्रियोल्स है, ठीक है, और क्रोमाटिन है, वो टाइटली कॉइल्ड होते हैं क्रोमोसोम्स, ठीक है. सेंट्रियोल्स दोनों तरफ जाते हैं, और ये जो डीएनए है, डीएनए के क्रोमोसोम्स अगर हम देखें, ये क्रोमोसोम्स के क्रोमाटिन जो है वो बहुत टाइटली कॉइल्ड हुए होते हैं, ठीक है. लेकिन जब भी न्यूक्लियर डेवलप करता है तो वो ब्रेक करते हैं. ओके, एंड दैट टू सेंट्रियोल्स विल बी माइग्रेटेड इनटू ऑपोजिट डायरेक्शन. ओके, और ऑपोजिट एंड्स, ऐसे ये सेंट्रियोल इधर पे चलाएगा, ठीक है. अभी मेटाफेज में ये कंप्लीटली दो साइड आ जाता है और ये अटैच हो जाएगा अभी क्रोमोसोम से, ठीक है। उसके बाद ये क्रोमोसोम्स को खींचने लगेगा ताकि ये दो हिस्सों में बट जाए, ठीक है। समझ में आ रहा है ना? कोई भी क्वेश्चन हो तो मुझसे पूछ सकते हो, ठीक है। यू गाइस कैन आस्क एनी क्वेश्चन इफ यू हैव एनी डाउट रिगार्डिंग दिस टॉपिक। ओके, सो ये अब क्या होता है, अब एनाफेज में ये डिवाइड होने स्टार्ट हो जाते हैं। लास्टली जब टेलोफेज आता है वो कंप्लीटली क्रोमोसोम्स को डिवाइड कर देता है और दोनों डिफरेंट न्यूक्लियाई पैदा कर देगा, ठीक है। सो दैट इज माइटोसिस, ओके, इन इंट्राफेज, प्रोफेज, मेटाफेज, एनाफेज एंड टेलोफेज। ओके, दिस इज द phases of mitosis. Now we will move on to the meiosis. So meiosis is what? Meiosis is the process of reduction of division in which the number of the chromosomes per cell will cut into half through the separation of the homologous chromosome in a diploid cell. Okay, अभी अगर देखो, अभी अगर ये डिप्लॉयड सेल है तो इससे पहले तो दो सेल डिवाइड होंगे, दो न्यूक्लियाई होंगे जो कि एन होंगे, ठीक है। इट विल बी एन। अगेन फ्रॉम दैट टू सेल्स विल बी divided, again that will make 4. Okay, from each cell they will make two daughter cell and they will make total 4 numbers of daughter cell. Okay, so that is the meiosis process. Now this meiosis process have been divided into 2 parts. Okay, पहले फेज में क्या होता है, homologous chromosome pair at metaphase plate and migrate the opposite poles. meiosis 2 में वही chromosomes spread होंगे और they will be across over the metaphase plate and the sister chromatids will separate and migrate the 2 opposite poles. Okay, and they will make the daughter cell. There also we have prophase and all. Okay, so next we will move on to the cell. As we were discussing what were the cell, first we will know what is the definition of cell. So cell is a generally defined as the basic structural function and biological unit of from all form of life. Okay, आप अगर कोई प्लांट देख लो, ठीक है, कोई प्लांट, ओके, माइक्रो ऑर्गेनिज्म, ह्यूमंस, then if we say any kind of animals, okay, or if we also say like arthropods, okay, anything, okay, anything that have life is made with cells, maybe a singular cell or maybe bunch of cells, thousands and millions of cells. Okay, so that is the definition of cell. It is generally defined as a basic structural and functional and biological unit of all form of life. Okay, so it is capable of performing the functions of life, hence it is known as the fundamental unit of life. Remember that this is a cell is a fundamental unit of life. Okay, the study of cells is called as biology. Okay, you may have heard in your class 10, okay, class 10 or class 12, 11 who have studied with science and all, but still if you have not studied with science background also, you may have known there is different part called as biology. Okay, so the cell together, okay, when they will club together they will make tissue. Okay, then the tissue will form organs, different types of organs like our brain, kidney, stomach, lungs, heart, liver and all. Okay, then the organs will make a system. Okay, and that system will make human body. Okay, so let us know what are the functions of a cell. The first and the foremost function the cell performs will following function is provide the structure to the body. If you see, this is my hand, how this structure is. Okay, I have 4 fingers. Okay, my palm is like that, it is smooth. Okay, or maybe my nails are rough. Okay, so this structure, how this structure will be formed, this is formed with the help of cells, a bunch of millions of cells. Okay, so it take nutrients from the food taken through oral cavity, whatever we are taking orally, from that the cells are taking the nutrients to grow themselves. Okay, then it converts the nutrients into energy. It is not only absorbing but also it is converting the food, okay, It is converting the food into energy. Okay, energy. Also, it will carry out specialized function of the body. Okay, it has the body hereditary material that can make its own copies. Okay, so these are the main function of a cell. Now we will see the cell structure. So if we see in detail, there are cell membrane, which is the outermost surface of a cell, cytoplasm, which is the intracellular fluid which is packed with organelles. Okay, organelles like mitochondria, ribosomes, centrioles, Golgi body, Golgi apparatus. Okay, then also nucleus and all. Okay, so that is cytoplasm. Then we have nucleus, which is an organelle that controls the cellular activity. Whatever the chemical activity or physical activity, whatever is going on over there, it will be controlled by the nucleus. Okay, so you can see here, this is the cell membrane. Then we have mitochondria, which is the powerhouse of the cell. Then we have DNA, okay, and nucleus. Then we have endoplasmic reticulum, which will provide the structure for the cell. Then lysosome, which will engulf the pathogenic microorganisms or any foreign body. Okay, then we have ribosome, which will provide protein and transport to synthesize different types of food, okay, nutrients and all that also help. Then we have Golgi apparatus. Okay, so this is the main structure. Okay, now we will move on to the membrane. What is the function of the membrane? So membrane are actually the thin sheets of tissue. If you see in the brain, okay, if you see the brain, in brain also, okay, if you see the brain, that brain and spinal cord is also covered with a tissue layer, a thin layer of tissue, that is called as what? Can you guys guess? Okay, what is the name of the brain membrane? You can write it down to the comment section. Okay, so the name of the brain membrane is meninges. Okay, and for the heart it is myocardium. Okay, so there are different types of membrane. Okay, so what is the function of this kind of membranes? The first one is it will cover the surface. Second one, it will divide partitions. Okay, if you have different organs, okay, then it will divide in partitions. Then it will line a hollow organ or a body cavity to store fluids and all. Okay, that is the main function. So it have categorized into two types: epithelial membrane and connective tissue membrane. Epithelial membranes are serous membrane or mucous membrane, but connective tissue membrane are synovial membrane and meninges. Okay, so the epithelial membrane is consist of epithelial tissue and connective tissue which it is attached. Okay, so the serous membrane or mesothelium, okay, sorry for the pronunciation, the serous membrane extremely sorry, the serous membrane or mesothelium is the cavities of internal organs. Okay, it comprises of two layers. One is parietal layer, one is visceral layer. The parietal layer will be line body cavities, and visceral layer it will lines the internal organs. So what is the function of this? The function is to secrete a thin watery fluid. Okay, thin watery fluid which is called as serous fluid, that will decrease the friction in between the organ during the movement. Okay, like for the brain, okay, for the lungs and all. Okay, example as pleura, which is for lungs, serous pericardium, and we have peritoneum. Okay, so these are different types of epithelial membrane. Next is mucous membrane or mucosa, which secretes the mucus, a thicky sticky substance. It is composed of an epithelium layer which will be lining over a loose connective tissue. Okay, and from the extensive continuous lining, the organs are connected with the outside of the body, such as digestive, respiratory, urinary and reproductive system. So what is the difference here? In the system, The acar mucosa or mucous membrane are not only being covered the system, but also they are attaching the inner organs to the outer side of the body as well, okay, like digestive, respiratory, urinary and reproductive system. Next we have functions that depends on location, like for respiratory the mucus traps over there will be trapping the particles. Cilia will force a particle to out of the body. Like say whenever we will go in very dusty place, we will start sneezing, okay, or we sometimes we have runny nose, that mucus, our mucus inside the nose, nasopharynx and all, or in our throat also it is there, okay, it will trap the particles and microorganism ताकि वो अंदर ना जा सके और हमें कोई बीमारी ना हो, ठीक है ना. फिर डाइजेस्टिव ट्रैक्ट में भी ये होता है, और ये म्यूकस प्रोटेक्ट करता है ऑर्गन को स्टोमैक एसिड से. अभी देखो अगर हमें तो पता है लाइक हमारे स्टोमैक जो एसिड है, हाइलुरोनिक एसिड जो HCl जो स्टोमैक प्रोड्यूस करता है, उसका pH 3 से 4 के अंदर होगा. अभी अगर कोई देखो, अभी अब अगर आप स्किन के ऊपर एसिड फेंको तो वो जल जाएगा, है ना. तो इसलिए भगवान ने बनाए ऐसे हमारे स्टोमक को ताकि ये एसिड तो प्रोड्यूस कर सके बट जले ना. वो कैसे? टू विद द हेल्प ऑफ द म्यूकस प्रोटेक्टिंग लेयर. ओके, अंडरस्टूड, राइट. Next is connective tissue membrane, which have synovial membranes. You know where is synovial membranes, right? In the joints, in every in the knee joints. Okay, especially in the knee joints, you can see the synovial membranes. Then meninges, which is I told in the brain. Okay, the outer layer of brains. Next we have glands. So what is the definition of a gland? A gland is an organoid that is specialized to produce a substance that is sent out to other parts of the body. अभी organ और एक gland में क्या difference है? ये organ ये बनता है इसलिए ताकि इससे कोई एक एंजाइम पकड़ो, ओके, एंजाइम, हार्मोन या तो फिर कोई भी एसिड, ओके, कुछ भी केमिकल रिएक्शन ये ग्लैंड है ना, ये ग्लैंड रिलीज करेगा और ये दूसरे बॉडी पार्ट्स या फिर दूसरे ऑर्गन्स में भेजेगा ताकि उनका काम हो सके, ठीक है. सो दैट इज व्हाट अ ग्लैंड इज. ओके, सो नाउ ग्लैंड्स आर आल्सो डिवाइडेड इनटू टू. ओके, फर्स्ट वन इज exocrine glands and then we have as endocrine glands. Okay, so what are the exocrine glands are there? So we have the tubes or ducts to carry the secretion away from the glands. Okay, they may be unicellular or multicellular. In unicellular we have the single cell, example as goblet cells. Okay, and multicellular we can say there are many secretory cells as categorized, such as simple tubular, branched tubular, then coiled tubular, simple sickle cell and branched acinar. Okay, this one you can see in lungs. Okay, so these are the types of exocrine glands. Okay, then the example is the gastrointestinal glands are secretes digestive juices. Okay, then subcutaneous glands will produce the oil or sebum to protect the skin and to protect the moisture of the skin. Okay, then lacrimal glands which will produce tear, which you can find here in the inner corner of your eyes. Okay, so these are the examples. Next we will move on to the endocrine glands. The ductless glands are secretes directly into the blood, so they are not producing some secretion and then they are moving it towards others. No, they will directly mixed with blood, they will release the secretion directly into the blood, and the hormones have effects on specific target tissues such as pituitary gland, thyroid gland, adrenaline gland. Okay, LHH, FSH, TSH. Okay, different types of many hormones are there. Okay, now as we have learned, it is that is the exocrine gland. Our stomach is an exocrine gland and our stomach will release gastric juices. So what is this mechanism of gastric juices? First one, we know that the first and most important gastric juice is HCl or hydrochloric acid. Okay, so hydrochloric acid, what it will do? It is secreted in the canaliculi of the parietal cells, and HK-ATPase pumps the H ions actively in the gastric lumen. So what will happen? It will be divided and H2O and CO2 will be formed. That H2O or CO2 will be produce the carbonic anhydrase and it will move forward to the acetylcholine. Then the histamine will be act, okay, it will act and it will produce the H2CO3 hydroxy carbon dioxide. Then it will be divided into two. One is H active, one is H positive ion, and one H carbonate ion, okay, hydrocarbon ion, which is HCO3 minus, okay, and it will also produce the ADP, okay. So the gastric secretion, first one is the pepsinogen secretion, where the secreted from the chief cell, okay. So pepsinogen secretes from the chief cell. अभी ये क्या होगा, ये जो पेप्सिनोजन secrete हुआ है ये synthesize होगा, okay, like protein, अदर प्रोटीन की जैसे ये भी synthesize होगा, फिर ये जाके स्टोर होगा जाइगोमेन ग्रेन्यूल्स में. उसके बाद यही पेप्सिनोजन कन्वर्ट होके पेप्सिन में बदलेगा. वो किसके हेल्प के? वो HCl के हेल्प से, ठीक है. फिर ये पेप्सिन स्ट्रॉन्ग होगा पेप्ट्रोलाइटिक एंजाइम्स में और वही पेप्सिनोजन सिक्रेट होके स्टिमुलेट करेगा गैस्ट्रिन और हिस्टामिन को ताकि HCl काम कर सके, ठीक है ना. नेक्स्ट है म्यूकस सिक्रेशन. अभी ये म्यूकस कैसे सिक्रेट होता है? ये secreted by mucus secreting cells, okay, and these are form a protective layer in gastroepithelium, okay, to protect it from the low pH of the gastric juices. Then it stimulated by increasing blood flow in the stomach, okay. So this instinct factor secretion also include the secreted from the parietal cells along with the HCl. It is a glycoprotein which will help vitamin B12 to absorb from the terminal ileum, okay. So that is the mechanism. Now we'll move on to the regulation of gastric secretion. First one is the neural secretion, okay, neural regulation. Now this is regulated by the neural and hormonal mechanism, okay. If you see here, okay, the local autonomic reflexes will be involving, okay, it will be involving the cholinergic neurons which will transfer the impulses from the central nervous system by a way of vagus nerve, okay, and that vagus nerve will cause direct stimulation and secretion of the gastric juices, okay. So as an example I can tell, see, daily you are eating, you are having your breakfast on 8 o'clock in the morning, okay, 8 a.m. in the morning. Now one day, just for one day you didn't had food, okay, you didn't had food till to the 12 p.m. Now what will happen? Now you have a practice of having the food 8 a.m. so your brain have been trained like that. The vagus nerve will release some impulses just at 8 o'clock or around that, and that will make your stomach to release those gases. But now you are not having, so that HCl gas will still remaining released by the cells, okay. So this is kind of an example of the neural regulation, okay. Next we have factors that stimulate the gastric acid secretion. What are those? Acetylcholine, gastrin and histamine. The factors that inhibit the gastric acid secretion include pH of gastric lumen, somatostatin and chyme in duodenum, okay. The regulation of gastric secretion, okay. So it is mediated by the release of hormone gastrin. The gastric secretion occurs in three phases. First one is cephalic phase, then we have gastric phase, and third one is intestinal phase. And each of the phase have different types of mechanism, okay. So this is all about the gastrointestinal system. Next we will move on to the next system, that is musculoskeletal system, okay. So in the skeleton we have different parts, okay. So if we see, these are cranial bones, okay. Here we have our humerus, radius, ulnar, phalanges, carpal, metacarpal. Okay. Then we have rib cage. This is the sternum. This is our spinal cord. Here we have our pelvis. In the back side of this we have scapula. You can see over here, this is scapula. Okay. And we have femur, and outside of that we also have radius, ulnar, tarsal, metatarsal and phalanges. Okay. So this is the main skeletal system. अभी स्केलेटन सिस्टम को अगर हम दो हिस्सों में बांटे तो ये दो टाइप के स्केलेटन हो जाए. एक है एक्सियल स्केलेटन. ठीक है. अभी एक्सियल स्केलेटन में क्या क्या आता है? एक्सियल स्केलेटन में आता है ये ऑसिकल्स, इनर ईयर, हाइडॉइड बोन. ओके. फिर ये पूरा रिब केज, स्टर्नम और ये वाला पूरा वर्टेब्रा वाला पार्ट आ जाएगा. पूरा आपका फेस, ये स्कल जो है ना, ये पूरा आएगा. सो द एक्सियल स्केलेटन एक्सिस ऑफ द बॉडी कंसिस्ट ऑफ द स्कल, वर्टेब्रा कॉलम, स्टर्नम और ब्रेस्ट बोन एंड इंक्लूडिंग रिब्स. Okay, now we have appendicular skeleton, which includes our clavicle bone, scapula, humerus, radius, ulnar. Then we have carpal, metacarpal phalanges. Then we have the pelvic girdle, then femurs, tibia, fibula, phalanges, tarsal, metatarsals. Okay, so these are the bones over there in the appendicular skeleton system. The appendicular skeleton consists of the shoulder girdles and the upper limbs, and the pelvic girdle and the lower limbs. Okay, so that is all about the skeleton system. Why I am not going in detail? Because you guys have to prepare a bit for, and this is the most important topics. Okay, don't forget, whatever you know, try to write it, and whenever you are writing, don't write it too broad, don't write it too short. Okay, see, if there will be paper, right, exam paper, so whenever you are writing, little bit space you leave here, little bit space over the sides. Okay, very little, not too much. Okay, examiner should not think, उनको समझ में ये नहीं आना चाहिए हाँ इसने जानबूझ के ये स्पेस छोड़ा है ताकि आंसर इतना ज्यादा ना लिख सके कि इसको पता ही नहीं है. ऐसा मत करो. ठीक है, थोड़ा सा स्पेस छोड़ दो. Always start with an introduction, then go for definition, then go for detail, then you can provide the pictures over there as well. Okay, so let's start with the system of the human body. First one is cardiovascular system. Okay, so what cardiovascular system have inside that? It have heart, blood vessels and blood. The major function of the cardiovascular system is to transport of material throughout the body. Okay, now if we see in the structure, this is superior vena cava, this is inferior vena cava. Then we have tricuspid, bicuspid and mitral valve over here. Right atrium, right ventricle, left atrium, left ventricle. Okay, so these four ventricles are here. Then we have aorta or aortic arch. Okay, pulmonary artery. Okay, so this is the part of cardiovascular system. So what is the function of heart? The first and the main function is to pumping the blood. Okay, and it will take the deoxygenated blood and make it into the oxygenated blood. With the cardiovascular system, the respiratory system is also interrelated. Okay, the blood vessels, mainly all of the blood vessels will carry out the deoxygenated blood. Okay, deoxygenated blood, or we can also say carbon dioxide blood. Okay, then we will come to arteries, which will carry the artery will carry the oxygenated blood, okay, to all the cells, tissues and organs of the body. Okay, so that is the main function of cardiovascular system. Next, move on to the digestive system. Now digestive system include GI tract, liver and pancreas. So we will start with the upper digestive tract, that includes mouth, okay, cavity, okay, then we have tongue, esophagus, okay, esophagus will travel down from here. It will start with the stomach, then we have large intestine, small intestine, colon, rectum and anus. Okay, so this is the main digestive tract. Now first, what your mouth will do. Mouth is a hollow muscular organ because it's including our muscles over the jaw, then muscles of our cheeks and also the tongue. Okay, it consists of the palate, teeth, gum, okay, then we have tongue. Okay, so what is the function? Mainly in the mouth there will be chewing, like mastication. ओके, खाना चबाते हैं या खाना को सलाइवा के साथ मिक्स करते हैं, जो भी होगा ये सब माउथ के अंदर होगा. फिर उसके बाद ये ही खाना और ये बोलस, ठीक है, जाएगा ईसोफेगस में, ट्राकिया से ईसोफेगस में जाएगा. ईसोफेगस में जाके ये क्या होगा, ये थोड़े से एंजाइम के साथ मिक्स होगा और ये ट्रैवल डाउन करेगा स्टोमक की तरफ. स्टोमक में बहुत सारे डाइजेशन प्रोसेस होंगे, डिफरेंट टाइप के न्यूट्रिएंट्स के डिफरेंट डाइजेशंस. ओके, अभी ये डाइजेशन के बाद जो भी प्रोडक्ट अच्छा है, लाइक विच योर बॉडी नीड, दैट योर बॉडी विल कीप और एब्सॉर्ब. अदर प्रोडक्ट दे विल ट्रांसफॉर्म इट टू द स्मॉल इंटेस्टाइन. अभी स्मॉल इंटेस्टाइन में ज़्यादातर लाइक 60% to 70% of the nutrients will be absorbed in the small intestine. Then other than that, whatever the waste product is there, again it will go into the large intestine. उधर भी थोड़ा स्टोर होता है और बाकी जो भी बचा हुआ रहता है वो सारे products, okay, it will be outside of the rectum, okay, through the anus. So that is GI tract. Next we have liver and pancreas. This the pancreas sometimes it will not sometimes it will work as a release of bile ducts and all. Also sometimes gallbladder is also included in the digestive system. So next we have is system of the human body. Okay, other system what we have, endocrine system. So as we were discussing, endocrine system includes different endocrine gland. What are the glands? They are hypothalamus, pineal gland, okay, thymus, trachea, then heart, stomach, kidney, skin, lung, liver, adrenaline gland, pancreas, small intestine, testis, everything is an endocrine gland. Okay, so the major functions are to coordination of the function of the body through release of regulatory hormones. It will also, that hormones will target different kind of different kind of organs and make them to work for particular time of period. Okay, so that is the function of endocrine system. Then we have immune system. What is immune system? Later we will, in microbiology we will discuss it later. Okay, so in immune system we have thymus, spleen, lymphatic system and WBC or white blood cells. Okay, just give me 1 second. Yeah, okay, so immune system is include of thymus gland, spleen, lymphatic system and white blood cells. The major function is to defense against the microorganism. Other than that, we have integumentary system with the components of skin, which will like work as a protection against the external environment. Then musculoskeletal system, which components are skeleton, muscles and bones. The major function is to movement and the support. Then we have nervous system, which includes components such as brain, spinal cord, peripheral nerves, and the major functions include coordination of the body function through the electrical signals and release the regulatory nucleus cognition. Okay. Then in the reproductive system we have gonads, penis, vagina and uterus. The major function is the procreation or the reproduction. Okay. Lastly we have respiratory system, which components include lungs. Major functions are oxygen and carbon dioxide exchange with the environment. Okay. So that is the main function of the respiratory system. Next, let's move to the microbiology. Okay, so let's get just revised one. So what are the important that can come? Note it down. So there are 12 systems on the body. Okay, other than that, cell structure. Before starting the microbiology, note down all the important questions that I am telling. Cell structure, cell membrane, then we have fluid mosaic model. This is the first unit. In second unit, in the tissues, classification of tissue, also classification of epithelial tissue. This is the most common question. Okay, then we have the function and structure of. Now what are the organs mainly? Note it down: heart, kidney, lungs, nephron, brain. Okay, these are the most common ones, and especially for the physiology. For the physiology, blood circulation, mechanism of breathing, urine formation, then also the negative feedback. What is negative feedback? It can come. Okay, so from this only your questions will come for anatomy and physiology. Try to read out this question. I am guaranteeing you that at least 40 to 45 marks of questions will come from this. It will definitely come from this. Okay, so don't forget to read out those. Okay, next we will move on to the microbiology. So let me just wipe it out. Okay, so microbiology. So what is microbiology? Microbiology is nothing but it is a study of microorganisms and human health disease which are only visible under the microscope. To learn the bacteria, we will call it as bacteriology. To study about the bacteria virus, we will call that as virology. Study of parasite is parasitology. Study of fungi is mycology. Lastly, study of study of fungi, that is mycology. Okay, next just move on some some greater scientists who have been invested in the microbiology. So the Joseph Lister, who are born between 1827 to 1912, what he have done? He is called as the father of the antiseptic surgery. क्यों? क्योंकि वही फर्स्ट है जिन्होंने की ओके पोस्ट ऑपरेटिव इंफेक्शन के बारे में रिसर्च किया है और तरीका ढूंढा है कैसे. ही हैव पोस्ट्यूल्ड दैट पोस्ट ऑपरेटिव इन्फेक्शन कैन बी रिड्यूस्ड बाय यूजिंग डिसइंफेक्टेंट्स टू स्टेरिलाइज द सर्जिकल इंस्ट्रूमेंट एंड टू क्लीन द वुंड. Okay, next one is his antiseptic surgery involves the carbolic acid. Okay, then it comes Robert Koch. You may have heard for 100 times. Okay, he have been born between 1843 and lived till the 1910th. Okay, he is called as father of bacteriology. Sometimes for 1 mark question, fill in the blanks, or for to true or false also it can come. Okay, don't forget this. He is the father of bacteriology who introduced the staining technique to recognize microorganisms under the microscope. Also hanging drop method he have introduced, discovered different bacteria such as anthrax bacillus, tuberculosis bacillus and vibrio cholera. Excuse me guys. Yeah, next we have is Koch pustules. What he have proved that microorganisms can be accepted as a causative agent of an infection disease only if the following condition is fulfilled. What is that? The microorganism should be constantly associated with the lesion of the disease. Okay, wherever it's happening it should have to be associated. Then it should be possible to isolate organism in pure culture from the lesions of the disease. Then the same disease must result when the isolated microorganism is inoculated into the suitable laboratory animal. Okay, lastly it should be possible to re-isolate the organisms in pure culture from the lesion produced in the experimental animals. So that are the Koch pustules. Now I'll move on to the other important contributors such as Paul Ehrlich, who is the father of chemotherapy, proposed the side chain theory and discovered the salvation. Then Edward Jenner, who developed the first vaccine in the world, which is smallpox vaccine. Okay, that Edward Jenner question also sometimes come. Okay, so try to remember that as well. So biological classification of taxonomy method, classifying and categorize the living organism into groups similar. They are shared similar characteristics and features. So the major classification of a microorganism are bacteria, virus, fungi, protozoa, algae. Okay, so if we see each of these microorganisms, they are divided into two types of cell: prokaryote and eukaryotes. Eukaryotes are nothing but they are nucleus bounded cell. Okay, and the nucleus is bounded with nucleus membrane, where in the prokaryote cell or older version of cell we can also say it don't have any kind of nuclei or lack of nuclei, whatever. If the nuclei is present also, that will be very weak. It have multiple diploid linear chromosome, but whereas in the prokaryote cell it have only single cellular chromosome. Eukaryote cell have 80S ribosome and prokaryote cell have 70S ribosome. The eukaryote cell membrane, the bound cell, it will bound the cell organelles such as vacuoles, mitochondria. But in the prokaryote, lack of membranes will bound organelles such as vacuoles and mitochondria, etc. The examples of eukaryotic cells are fungi, algae, helminthes and protozoa. And as an example of prokaryote cell, I can say bacteria, different types of bacteria, chlamydia, mycoplasma, spherococcus and all. So this is the main difference between eukaryotic and prokaryotic cell. Okay, this is very important question. This is a very important question. Okay, it comes for 5 marks or 2 marks, 4 marks, like that. Okay, so don't forget. Then we have staining. So we know what is staining. To know that staining is a procedure where we are pouring different types of staining or colors to identify the microorganism under the microscope. Okay, so what is staining? Staining refers to the dye or colored fluorescent substance that is applied to biological specimen to enhance their visibility and constant under a microscope. The stains will contain different types of dyes or fluorescent molecule, so that we can recognize them under the microscope. Okay, so there are different types of staining: gram staining, okay, simple staining, acid fast staining and all. So we have simple staining, differential staining, lactophenol staining, cotton blue staining, or KOH mount. Okay, so first let's know about the simple staining. In simple staining, a single dye stain, okay, a single stain or dye will be applied into the specimen, which uniformly colors only the cells or structure that is present in that culture. Okay, what are the color that we can use methylene blue, crystal violet, and saffronin, or which will perform the pink or red color. This technique will primarily use to visualize the morphology, size and arrangement of the cells under a microscope. Okay, let's move on to the next one. Positive staining. Okay, so the bacterial skull that are stained directly while the background remains colorless. Okay, example as crystal violet and methylene blue. Okay, you can see over here the change of the positive staining. Then negative staining, which include the microbial, in which the microbial background is stained directly while the bacterial cells remain colorless. Okay, the acid stain are used as India link and nigrosin. Okay, so you can see here the structure over here are not get colored yet, but whereas the background got stained, so that is called as negative staining. Then we have differential staining. So differential staining techniques are used to identify multiple microorganisms at a time. It is used multiple dyes to distinguish between different types of cells or structure based on their chemical or physical properties. Okay, so they impart different colors in different bacteria. Most commonly employed different stains. Okay, so most commonly are gram stain and acid fast stain. Okay, so what is gram stain? It is the most widely used stain in the bacteriology to identify different types of bacteria. It helps to differentiate gram positive and gram negative bacteria based on the properties of their cell walls. Okay, so the gram stain, how to do a gram stain? To do a gram stain, we have to maintain this principle. Okay, gram stain also again a very important. It is a very important topic. Okay, questions can come. So it involves staining bacterial cells with the crystal violet dye, iodine solution, alcohol and a constraint such as saffronin. The gram positive bacteria retain the crystal violet stain due to their thick peptidoglycan layer in their cell wall and which traps the dye, whereas the bacteria appear purple under microscope. For the gram negative bacteria, they don't have any kind of membrane or peptidoglycan layer, so when you wash them with alcohol, they will lose this violet dye. Okay, and whenever again you will try to put the saffronin dye, what will happen? The gram positive bacteria have a thickened layer, so he will not absorb it, but this gram negative bacteria will easily absorb the color, so that we will able to differentiate the gram positive and gram negative bacteria under the microscope. So first one, what we have to do is we have to heat fix the smear specimen and stained with crystal violet dye for 1 minute. Okay, pour the gram iodine, which will dilute in the solution of iodine, over slide for 1 minute. Okay, then the decolorization or acetone for 10 to 30 second alcohol can be used. Okay, then that will counter stain with dye of saffronin. So what will happen? It will perform two different colors. Okay, the one who have the purple stain will call it as gram positive, and the one with pink or red stain will call it as negative, gram negative bacteria. Okay, next we will move on to the acid fast staining. So acid fast staining is also called as Zale-Neslin staining. This is also a very important question. 4 marks or 2 marks can come from here. Okay, so acid fast staining is actually a carbon fusion is produced in one slide containing fixed with smear, left for 5 to 10 minutes. Increment heat is given to the slide. Okay, and after that what will happen? It will wash under the tap water. The stained smear is decolorized 25% of sulfuric acid over 2 to 4 minutes and washed under with water. Okay, so that counter strain of 2% methylene blue is for, we will do it for 30 seconds. Then with water, and here we will wash and dry. Okay, so what's happening in the first, we are stained with 25% of sulfuric with the carbon fuchsin. Okay, carbon fuchsin. Then it will stain all the cells in red colors. Then again what we are doing, we are putting the sulfuric acid. So except these bacteria, other bacteria will be stain free. Then again we are putting methylene blue. So because they have tough peptidoglycan layer outside of their cell, they will not able to absorb this methylene blue, but the other cells, okay, will be absorbing the methylene blue. Lastly, we have the acid fast bacteria, which is pink or red color, and the non-acid fast bacteria will be in blue color. Okay, so next we will see the bacterial growth or curve. The bacterial growth of curve typically follows a pattern known as growth curve, which consists of several phases such as lag phases, including metabolic activity without division, then log phase, which will rapid cell division. Then we have stationary phase, there will be phase of equilibrium, and decline phase, which will be death phase. So if you see if I draw, okay, this is the growth of the bacteria. Wait, let me just write it so that you guys will able to understand. So see, if it is a graph, okay, and this is the growth of bacteria and this is the time. So what we can see here, first there will be lag phase, which will be very slow. Then there will be rapid cell division and the growth will be sudden up. Then we have phase of stationary phase, where it will be equilibrium or the growth will be normal or continuous. Next we have decline phase, where rapidly cell death will be there. Okay, so this is the bacterial growth curve. Now it is dependent on different types like as temperature, pH, environment, moisture and all. So what is the nutrition of microorganism? All living organisms require a source of energy to survive. Depending on the source of energy utilized, microbes are classified as phototrophs, chromotrophs, lithotrophs and heterotrophs. Lastly we have autotrophs. So phototrophs are cyanobacteria, chemotrophs are nitrobacteria, lithotrophs are purple sulfur bacteria, heterotrophs are E. coli and autotrophs are green bacteria. So these are just an example. Okay, so what is the ultrastructure of bacterial cells? So as we were seeing, that is mainly the bacterial cells are prokaryotic cells. Okay, so this will made with capsule, cell wall, plasma membrane, mesosome, cytoplasm, nuclear, DNA, flagellum, plasmid inclusion, polyribosome and pilus. Okay, or pili or fimdi also we can say. Next we will move on to the immunity. Okay, from immunity also 5 to 10 marks question can come. What is mean by immunity? What are the classification of immunity, or what is the classification of acquired immunity? Describe in detail. Okay, so immunity is refers to the resistance exhibited by the host towards the injury that caused by microorganisms and their product. Okay, toxin products. Okay, so we have three types of immunity, that are innate immunity, adaptive acquired immunity, or herd immunity. What is innate immunity? First, let us know about the herd immunity. Herd immunity is what? See, if you are a person, okay, you are not vaccinated, okay, but the people around you are vaccinated. So what will happen if there is someone who is already affected? This will not gonna spread to you because these people are not getting affected. Okay, you are protected by this layer. So that is called herd immunity. Then we have innate immunity, which can be specific and non-specific. The specific one includes skin, ciliary clearance, low stomach pH, and lysosome. The non-specific one include monocyte, eosinophils, basophils, lymphocytes, and neutrophils. Now, adaptive and acquired immunity, which can be active or which can be passive. Active immunity is natural, or it can be vaccinated. Like if you are getting one disease condition, again it will not happen because that antibody is already activated in your body. Okay, in active acquired immunity also there are two types that I told. One is natural and one is vaccinated with the vaccine. So whenever if you got vaccinated, whenever that particular microorganism is coming inside your body, it will be affected by the vaccine's antibody. Okay, so the next one is passive immunity, which again I have been divided into two types, that are maternal and immunoglobulin treatment. Maternal, which immunity babies or fetus or childs are getting from the mother, and then is immunoglobulin treatment or through the immunoglobulin therapy. So we have to know about the antigen if we want to understand about the immunity. Okay, so the antigen molecules are substances that are capable of stimulating an immune response in the body. Antigens can be foreign substances such as pathogen, viruses, toxins, molecules from the non-pathogenic sources, etc. Okay, so if it is your body, you have an immune system. Okay, if you have an immune system. Now a pathogenic microorganism, okay, so he is trying to entering into your body. So this microorganism or this particle or this particular toxic particle is called as antigen, and with the protein or with the with the response that will be with the system that it will be respond is called as antibody. Okay, so we have exogenous antigen, then endogenous antigen, autoantigens, and lastly heterophile antigens. So exoantigen, what are those? Exoantigens these are the antigens that will be originates from the outside of the body, such as it is found like on the pathogens like bacteria and viruses. Then we have endogenous, which the antigen which will originate from the body within those found the surface cell infected by the viruses or the intracellular pathogen. Lastly, we have autoantigens, which are diverse from the body own tissue, and the autoimmune disease also we can call, where the antibody will be affecting their own body, okay, own cells. Then heterophile, which these antigens can be stimulate the production of the antibodies that cross react with other unrelated antigens. For example, Epstein-Barr virus produces the heterophile antigen. Okay, then we have antibody. Now what are the antibody? Antibodies are immunoglobulins, are the protein that are generated by the beta cells when when they will encounter with the antigens. They are identified and bind to antigen and like flagging them for elimination by the immune system or directly neutralizing the harmful effect. So we have immunoglobulin A, immunoglobulin B, immunoglobulin immunoglobulin A, immunoglobulin M, immunoglobulin G, immunoglobulin E. So antibody and antigen have different type of reaction, but what will happen when antibody will affect its own body system, or because of some reaction it will be actively it will be actively in action. Okay, so we have different types of hypersensitivity, that is include hypersensitivity 1, which is also known as hypersensitivity, immediate hypersensitivity or allergic hypersensitivity. It is an exaggerated immune response that will triggers by release of immunoglobulin E. और ये क्या करता है, ये antibody responsible है to presence of the allergen. Okay, ये response कब करेगा, जबकि कोई allergic पैथोजेन या फिर एलर्जिक फॉरेन बॉडी अंदर घुसे, जैसे कि पोलन या डस्ट. ठीक है, दिस टाइप ऑफ रिएक्शन ऑकर्स रैपिडली, यूजुअली विद इन मिनट्स आवर्स आफ्टर द एक्सपोजर टू एलर्जन. सो वी हैव एंटीजन एंड IgE. नाउ व्हाट विल हैपन देयर, फर्स्ट they will produce the mast cell. Now the IgV will be attached. Okay, the pollen food will be come in the, and it will attach to the IgE or immunoglobulin E. Next, this immunoglobulin will be attacking the mast cell. Okay, now the mast cell will be released outside and it will start allergic reaction. So first one, the sensitization or first allergen exposure will be there, where IgE product and binding will be there. Then re-exposure or subsequent exposure. Then deregulation or activation, immediate reaction or mediator effects. Then we have systematic effects or anaphylactic reaction. Here we can get shock as well. Okay, in this phase we can get shock also. Lastly, the resolution or the intervention. Okay, so this is hypersensitivity 1. Next we have hypersensitivity 4. This is the most common question that have come for at least 3 to 4 times in last 5 or 10 years. Okay, so type 4 sensitivity, also known as the delayed type of hypersensitivity or DTH, or the cell mediated hypersensitivity. It is one of the 4 types of hypersensitivity reaction mediated by the immune system. Okay, so what will happen, the antigen will be attached with the CD4 cell, then it will release the cytoplasm, then macrophages will be present. The macrophages will be eating the foreign body. So the antigen entry, where the antigen will enter through the skin, lungs or mucous membrane. The antigen will present, the antigen will be process the APC or antigen presentation will be processes the antigen and present with the MHC class 2 molecules. Then the T cell activation will be there, where the CD4 cell or T cell will be recognize the complex and become activated. Then memory T cell will activate the in memory T cell, the activated T cell will be proliferated, differentiate between T1 cell, Th1 cell from the memory of T cell. Then in the re-exposure, the memory with Th1 cell are recognized as antigen upon the re-exposure or rapidly activated and release. It will release the cytokines. Lastly, the inflammation and the damage, where the cytokines will recruit the activated more immune cell, causing the inflammation and the tissue damage. Now this is the hypersensitivity reaction 4. Lastly, we have a serological test. So we have different types of serological test. Okay, why it is to? It is to diagnose different types of microorganisms, okay, or infection that have been, or the problem that have been occurring inside our body. First we have immunofluorescence test or IFA. Okay, it come for 2 marks, to try to remember that. Then it is used the fluorescent labeled antibodies to detect the presence of specific antigens or antibodies under a fluorescence microscope. Okay, the application it is used for the diagnosis autoimmune disease, infections such as syphilis and certain type of cancer. Then we have agglutination test, which will detect the pressure of the antibodies, presence of the antibodies or antigen by the clumping, which is called as agglutination for the particles, so that the lab examiner will able to recognize this under the microscope. The types are lattice agglutination, extremely sorry for that. Okay, so there are types, okay, which includes active lattice agglutination using latex bead, and heme agglutination, which will use the red blood cell. Application most commonly used for blood typing, detecting the bacterial infection and rheumatoid factors. Okay. Lastly, we have the complement fixation test, or CFT, which measures the ability of antibodies to fix the complement of in the presence of any kind of antigen. Okay. The use is this is used for diagnosing infections like syphilis and viral disease. We have different types of other serological tests as well, include the neutralizer test, which will measure the ability to antibodies to neutralize the infected or ineffectivity of a virus. Application is used for virology, confirm the viral infections and immunity status. Then flow cytometry, which uses for the fluorescent labeled antibodies to detect and measure the physical and the chemical particles of the cells or the particles in a fluid. Lastly, how to use that? We use it in a immunophenotyping for diagnosing leukemia, lymphomas, and for research different type of cancer cases usually use. Okay, flow cytometry. Lastly, we have immunodiffusion test, which will detect the antibodies antigen by their diffusion through a gel and their participation lines. Okay, they have two types, that is autotriclone double diffusion and radial immunodiffusion. Okay, the application is to use for the fungal infection and some autoimmune disease. Next, we have vaccine. So what is vaccine? Vaccines is a biological preparation that provides the active acquired immunity to a particular infection or disease. These are typically weakened or killed forms of microorganisms or toxins. Okay, so we have different types of vaccine. It includes live attended vaccines, inactivated vaccines, subunit vaccine, conjugated toxin, viral vector, and nucleic acid vaccines. Okay. Or now, how to maintain or how to store a vaccine? It is stored by cold chain. Okay, what is cold chain? The cold chain also come for 2 marks or 5 marks. Okay. The cold chain is a temperature which is control the supply of the chain system used to preserve the integrity and efficacy of the temperature sensitive products such as particularly vaccines and biologics from the point of the manufacture to the point of the administrator. So what will happen here? First, the manufacturing and packaging will be there. Okay, where the raw materials will be for the primary packaging, then the secondary packaging, tertiary packaging, and final packaging will be there, which is called as palletization. Okay, the vaccines are produced and packaged under the strict temperature control conditions. Okay, the storage and distribution. Okay, from there will be global transportation, regional transportation, cold storage warehouse, local transportation, and successful vaccination. How the storage and distribution will be there? After manufacturing, the vaccines are stored in temperature control facilities until the distribution. Okay, they are transported to distribution the centers, clinic, and healthcare facilities, including the temperature control method like refrigerated trucks or insulated containers. Lastly, the temperature monitoring. The temperature monitoring devices track vaccines temperature throughout the cold chain. Okay. Lastly, we have immunization schedule. Immunization schedule also come for 2 marks. So immunization schedules are outlined a recommended time sequence of vaccination for individuals for all ages. Okay, if we see here, at birth a baby will get BCG vaccine till the 1 year of age. Okay, and the amount, the dose will be 0.5 up to 1 month, and if it is beyond the 1 month of age, then it will be 0.1 ml. Okay, and if it is below 1 month, then it will be 0.05 ml. Then diluent, it have to be manufacturer supplied the diluent, and the route is intradermal route. Okay, in the upper arm left. Okay, then hepatitis B, it is also given at birth within 24 hours of the birth. The dose is 0.5 ml and it have to be given in the intramuscular. OPV 0 or the polio vaccine, it will be given at the birth within the first 15 days, 2 drops orally. Then OPV 1, 2, 3, it also has been given till to the 5 years of age, 2 drop orally. Okay, then pentavalent vaccine 1, 2, 3, which is diphtheria, pertussis, tetanus, hepatitis B and HIV virus. Okay, so the age is 6 weeks, 10 weeks and 14 weeks. Okay, so the maximum age is for up to 1 year. The dose is 0.5 ml. It don't need any diluent. Okay, but the route is intramuscular. Lastly, we have fractional IPV or inactivated polio vaccine, which have been given at the age of 6 and 14. Okay, 6 and 14 week and up to 1 year of age you can give it. Okay, the dose is 0.1 ml and it have been given through the intradermal. Okay, so these are the main topics I have discussed. These are very important topics. Whatever I have told, I may have not been covered all topics, but I have included there what are the topics that will definitely come. I am guaranteeing you that from there 80% questions will be come from anatomy, physiology, and whatever I have discussed now, from there only it will come. Okay, if you guys have not seen or attended the class, go in the YouTube, search for Easiness. You can find there our recorded live video. Go through that. I hope you guys will able to understand that. Okay, so thank you for this session. See you in the next session. Meet you up with guys in the next live video. Until then, keep learning and keep exploring with Easiness. Thank you.